Throughout this course, we have been introduced to multiple facets of the brain. Our group took a lot of time to consider what aspects this broad topic could cover, from talking about the physical, such as the parts, locations, and their purposes, science, the process of how the brain functions, chemicals, the multiple that are released and what each are, and even the brain's connection to the nervous system. This has autonomy over the body, which can also be impacted by us emotionally and even spiritually. Keeping these topics in mind, we have decided this class has already taught us much about the basics. Because of that, we have decided to fixate on a more interesting topic that may be of direct importance to many of us as college students. That is drug use. Today I will be covering a segment of this topic along with my three group partners, Jasmine Macias Ledesma, Michaela McCormick, Osiraisis Cardenas, and my name is Ana Teresa Lopez Pinzon. We are Group 2 and hope you enjoy our presentation. Now to the topic, how does marijuana use affect the brain? The first research article we chose to help us dive into our question is titled Long-Term Effects of Marijuana Use on the Brain. The participants included 48 confirmed stoners, meaning they had partaken in regular use about 4 times a week for the past 6 months, along with 62 age and sex-matched controls, meaning they were clean from marijuana use. With the first 48 participants, the demographics included mostly white people, then Latinos, some Native American and Black people, but no Asians. In turn, the 62 clean controls included mostly Latino and white people, some Native Americans and Black people, along with one Asian participant. Why there were more clean people taken into account was not necessarily shared. This was a study organized by the Proceedings of the National Academy of Sciences of the United States of America. It was also supported by the National Institute on Drug Abuse, in which they share the goal of the study was to characterize alterations in brain morphometry and determine potential downstream effects and connectivity as a result of chronic marijuana use. In order to measure these concepts, it was made sure none of the marijuana users, neither currently nor previously, ever suffered from psychosis, traumatic brain injury, or any neurological disorder. Meanwhile, the members of the control group tested negative to a urine drug screening. All 110 participants were involved in 3 different MRI scan processes. The first was a high-resolution T1 weighted image, along with complicated math for measuring gray matter volume. The second was a resting state functional MRI scan to measure functional connectivity of the brain. The third was a diffusion tensor imaging scan for measuring structural connectivity between brain regions via white matter tracks. They were also subject to the marijuana problem survey, in which they were posed 19 hypothetical situations they needed to rate a 0, 1, or 2, depending on how much of an issue they felt the problem, if they did experience it, created in their life. A general statistical linear model was applied to assess the contribution of chronic marijuana use on cognition, gray matter volume, functional connectivity, and structural connectivity. Ultimately, a few points were discovered. Chronic marijuana use is associated with complex neuroadaptive processes, and that onset and duration of use have unique effects on these processes. This means how early along in life someone begins consuming plays a role in how the effects play out. It was found that marijuana users have less bilateral orbitofrontal gyri volume, the part of the brain that affects the cognitive process of decision-making. Results also show marijuana users have higher functional connectivity in the orbitofrontal cortex network, as well as higher structural connectivity in tracts that innervate the orbitofrontal cortex. This was measured by fractional anisotropy, in which some evidence showcased how this segment of the brain initially increases, then decreases with protracted regular use. This may be correlated to initial versus chronic effects of marijuana use. The article states, exposure to THC leads to neural changes affecting diverse cognitive processes. Although some have reported decreases in regional brain volume, such as in the hippocampus, orbitofrontal cortex, amygdala, and striatum, others have reported increases in amygdala nucleus accumbens and cerebellar volumes in chronic marijuana users. Inconsistencies are attributed to methodological differences across studies. Whether these reductions in brain volume lead to downstream changes in brain organization and function, however, is still unknown. It is likely that connectivity changes could also be present, where alterations in brain volume are observed as a result of marijuana use.
A study from the National Library of Medicine that was published in 2023 explains how medical cannabis affects those who consume it, and gives insight into how THC and CBD affects both our mind and body. In this experiment, 51 participants from Florida were asked to document their experiences with medical marijuana. In total, there were 42 whites, 9 non-white, 27 males, and 24 females, and 17 with active use of cannabis, 8 that have used within 1 year, and 22 that have not used within 1 year. All are over the age of 18 as well. During this procedure, the participants were asked to complete a baseline survey to know their state and well-being prior to the use of medical cannabis. After this, they were to complete 3 to 4 weeks of ecological momentary assessments to study the participants' thoughts, feelings, and behaviors during use. This would be followed up with open-ended phone interviews at the end of the EMA period with the question, how effective do you think the medical cannabis treatment is for your condition? This gave them the opportunity to share their opinions as well as the benefits and side effects. The study would be completed with a 3-month follow-up survey, with them recounting their experiences and the effectiveness of the cannabis. By the end of the experiment, all 51 participants had reported they received the effectiveness of medical marijuana. More than half had said that their chronic pains had been reduced significantly, as many of the participants began using cannabis for their pains. They had said they noticed a decrease in the need for their medications, saw improvement in their physical health with less pains in their movements, and saw an increased cognitive function as a result. Examples of the benefits they saw were improvement in sleep quality and mobility.
Apart from these two studies and the class text, we used another source to provide us with more background information on the plant itself. In it, we found marijuana directly impacts our brain function, including memory, learning, attention, decision-making, coordination, emotion, and reaction time. It makes it harder to learn, stay focused, and to remember things. It also interrupts normal development of gray matter, increasing health risk. One huge component to these outcomes are the chemical interactions the brain receptors, brain neurotransmitters, and the substance components have with each other. On its own, the brain already releases a multitude of chemicals, such as serotonin, which the class text lets us know is important for emotional states, impulse control, and dreaming. Serotonin is also involved with sad or anxious moods, food cravings, and aggressive behavior. We also have dopamine, which produces the desire to act, creating motivation to feel rewarded. Loss of dopamine causes disturbances in motor function. Acetylcholine is another neurotransmitter. This one regulates motor control over muscles involved in complex mental processes such as attention, memory, sleeping, and learning.
Article 1 tells us that THC is the main psychoactive ingredient in the cannabis plant. THC binds to the cannabinoid receptors, which are ubiquitous to the brain. Cannabidiol, also known as CBD, has a broad pharmacological profile, interacting with receptors such as CB1, 5-HT1A, and TRPV1, which regulate fear and anxiety-related behaviors, and is a non-psychoactive cannabinoid that is well tolerated by humans across a wide dosage range without causing psychomotor slowing, negative mood effects, and abnormalities in vital signs. A recent naturalistic study revealed that cannabidiol, also known as CBD, prevented the memory-impairing effects of acute THC in humans. A change in the relative ratio of THC to cannabidiol in cannabis will profoundly alter effect of the cannabis on memory in human marijuana smokers. There were reports that pre-dosing with CBD or higher CBD content in cannabis might protect against some THC-induced deficits in verbal learning and memory. When it comes to the articles, there were both consensuses and controversies. Both view positive attributes to marijuana consumption, such as the higher functional and structural connectivity seen in Article 1. Medical aspects are majorly explored in Article 2, like using it to heal and relieve pain, as well as findings for increased cognitive abilities. However, the controversies are that Article 1 also has evidence of negative findings, like the reduced orbital frontal gyri volume. There are also multiple valid points made on the inconclusiveness of whether use is truly harmful or not, since too many factors may be in play to point out one causation for a number of effects. Article 2 also has many neutral responses, or people that had no effect with the use. Adding the third source helps us broaden our lens by informing us about the natural properties and presenting a few unbiased possibilities one may experience while consuming. In relation to this class's main text, we are informed of multiple neurotransmitters releasing chemicals in our brain, which alter our state of being. This is important to keep in mind because marijuana particles will bind to CB1 receptors and alter the natural brain process, meaning some kind of impact of how these are released. In the future, some aspects to consider would be 1. Different, bigger sample sizes. 2. Even more variety of research methods, although some consistency would also be good to compare stats over time. And number 3, keeping mental health in mind to look for both helpful and harmful aspects of marijuana. And here is our list of references. This concludes our Group 2 presentation focusing on the brain and how marijuana use affects it. Thank you for listening.